get going, everyone. So welcome to Diggin In. My name is Lindsay Randall, and I'm the host of this speaker series. Diggin In is a series of live presentations with archaeologists from around the country, co-sponsored by the Robert S. Peabody Institute of Archaeology and the Massachusetts Archaeological Society. Join us every other Wednesday at 1:30 p.m. Eastern Standard Time through November for all of our presentations. For a schedule of dates and the presenters, please visit us at the peabody.andover.edu or at the Massachusetts Archaeological Society's Facebook page. Viewers are going to be able to submit questions directly to me via the chat function on either the side of your Zoom screen or down at the bottom, depending on where that is located. At the conclusion of the presentations, we will give our speaker time to answer as many questions as they can, with the understanding that they might not be able to get to them all. And today we are excited to have Dr. Bill Farley joining us. Dr. Farley is an assistant professor in the Department of Anthropology at Southern Connecticut State University. He earned an MA from UMass Boston in 2012 and his PhD from the University of Connecticut in 2017. Dr. Farley is a historical archaeologist whose research is focused primarily on Native American and Euro-American interactions during the 17th century in New England. And he is also a trained archaeobotanist and has done botanical analysis on sites dating from the Paleo-Indian to the recent historical and from a number of regions around the world. Welcome, Dr. Farley. Thank you for joining us today.
Well, thank you. Let me share my screen here. And then we'll get you all set up here. There we go. How's that look? Does that look okay, Lindsay?
Yes, it does.
All right, thank you. Thank you so much for having me, Lindsay, for the invitation to talk. This is really an honor and a pleasure. I'm in my office for the first time in months because the internet here is much more reliable. And there's all boxes behind me, excuse that. But as Lindsay said, my name is Bill Farley. I'm a professor at Southern Connecticut State University, and I'm mostly these days a generalist historical archaeologist, and I mostly work in Connecticut. But I have had this, actually when I was at UMass Boston, I studied under Heather Trigg there, for anybody who knows her, is a brilliant archaeobotanist, and learned this skill of doing archaeobotany. And I've had the opportunity many times in my career to just get to work on these really cool projects I otherwise would have nothing to do with because of this technique that I learned. And so I figure a lot of people who are sort of archaeology adjacent, who are interested in archaeology, who know a little bit about archaeology, might not have that much information about these really cool subfields of archaeology. And archaeobotany is one of these. It is, you're going to see today, I'm going to kind of focus on the region that I work in and projects that I've been associated with, or at least have had to study pretty deeply because for my own research. But archaeobotany is a relevant sub-discipline of archaeology everywhere in the world. And archaeobotanists work on the biggest questions of archaeology. For instance, there are thousands of archaeobotanists, and I've done some work in this field, but working on the general study of the invention and introduction of agriculture, for instance. And of course, for those of you who are aware, the invention of agriculture and the adaptation of agrarian lifestyles had massive implications for people in every corner of the globe where it touched people. And archaeobotany is a huge part of that, right? So there are archaeologists working on just massively important archaeological questions around the world in this topic. So, but I mostly wanted to, as the name sort of suggests here, I want to do a couple of things. I want to tell you a little bit about the method of archaeology. So that is, what is archaeology and how do we actually do it? Very, very briefly, because we only have 25, 30 minutes together, so I can't go too deeply into this. And then ultimately, I want to get to how we use archaeobotany interpretively. So that is to say, how do we use this data source, which is evidence of plants in the past, to learn something about human culture, about how ancient peoples or not so ancient peoples lived their lives. And so that's where this sort of title comes from. So let me see if I can get this to, there we go. All right, so first of all, let's just define our most important term here, which is archaeobotany. What even is this study? So the best way to do this, we can do a little etymological study here of this term, archaeobotany, and really briefly, right, it breaks down into these two parts, archae meaning old or ancient, and then botany, of course, the scientific study of plants. That's fine. That's a fine definition, but it doesn't get us all the way there. So I'm going to introduce you to another term that is used to describe archaeobotany. I never use this term, frankly, because it's really a mouthful, and I think it's a little gatekeepery sometimes, which is why people have kind of settled on this archaeobotany title. But the other term that we sometimes use to describe ourselves is paleoethnobotany. And so if we break this term down, we've got paleo, which really means the same thing, old or ancient. We've got botany again, the scientific study of plants. But then we add this really important ethno in the middle, as in ethnography or ethnology or ethnographic study, right? This means cultural. It means relating to people and to people's cultures and behaviors. And so paleoethnobotany is thus the study of old or ancient plants as they intersect or relate to human beings and culture. And that's really what we're doing with archaeobotany. So that puts us on the same page of at least knowing what the heck we're talking about here. As you might guess, botany is a huge science. Plants are hugely varied. So this is really a big topic. So I'm going to break it down a little bit further as we go forward here. So first off, people and plants, a cultural intersection. I'm going to cheat this a little bit because unfortunately we can't meet in person. And I can't see you all face to face and take live questions from you and live answers from you like I'd love to. So when I teach this in class, I often do this little exercise, and we'll spend some time on this, like 15 or 20 minutes, this little exercise, all to drive home a single point. And that point is that plants are actually diffusely important in our lives. They are in every aspect of our lives. So I usually just ask my students to start naming places that they intersect with plants in their lives in any way. So first and foremost, I mean, obvious comes up is food, right? So plants are the majority of our diets, usually, in most cases. They make up, in most cultures, they make up the majority, the bulk of people's caloric intake. Not always, but usually. And they're always an important part of diet. So that's obvious, right? It's the first place people go, well, I eat lettuce in a salad yesterday, right? Okay, obviously great. But it really goes a lot deeper than that. Medicines, which are incredibly important culturally and certainly biologically as well, are often derived from plants. And this is both modern medicines like the stuff in your medicine cabinet and culturally specific medicinal practices through all of time. Plants are an incredibly important part of medicine, something archaeobotanists are often interested in. Tools, many of the tools and things that we use are made of plants. Wood is an obvious first thought, which of course derived from trees, but this is true of so many tools. Fuel. Much of our fuel is derived from plants. So obviously charcoal and fuel wood, which is the fuel of most societies through history, is plant derived. Other peoples use grasses and dung, which is collected from animals, but is mostly made up of plant material. And then of course even fossil fuels that we use mostly derive from very ancient plants that have turned into oil or coal or other things like that, right? So fuel is often plant derived. As religious symbols, most religions around the world have plants as an important aspect of their religious symbolism, including ancient religions, which often connect important grains and things like that with fertility or other important religious aspects, all the way up to, if we think of modern Christianity and the symbol of the wheat and the grape as such important parts of the Eucharist, right? And so this is true I'm certainly not a religious expert, but I know that this is true of many, many religions, that plants are important symbols there. So that's intensely cultural. Also landscapes, the way that we actually maintain our landscapes, the way we control our environment to encourage certain foods to grow, for aesthetic purposes and creating gardens as displays signs of status or symbols of wealth. People have used plants and plantscapes in their homes and around their homes, and so on ad infinitum. So this is like a tiny fraction, right? Like I said, well, oftentimes with students, we'll spend 20 minutes on filling an entire whiteboard with hundreds of different ideas of the different ways that we interact with plants. But the take-home message is that they're really in every aspect of our lives. So obviously, as archaeologists, we're interested in this, and so we have to think about how do we actually study that? How do we actually get at these such important things? So let me take a couple of minutes. Hope I'm not moving. Yeah, we're doing good. Keep track of the time. Let me spend a couple minutes then on the methodology for this. And I mean, this is like super brief. Like there's an incredible amount of, you could take a whole graduate course on this. In fact, I did. So this is to spend the next 2.5 minutes on this is going to be pretty limited, but just to give you a really quick introduction to the methodology of this. I'd say the big point I want to drive home here is that the reason why archaeobotany is a specialty and a subspecialization of archaeology is actually methodological mostly. The way we interpret archaeobotanical materials is not that different than we interpret any other kind of artifact. We think about them in the same ways, usually, theoretically. But methodologically, to actually collect botanical materials takes a very different approach, or a different set of approaches. So we rarely, sometimes, but rarely find botanical material just in like normal excavation, screening. You know, sometimes you'll get lucky and you'll get a seed or a piece, and charcoal often will be captured in regular screening. But the majority of botanical materials are recovered through different techniques. So let me start by separating out botanical analysis into our two big subcategories: microbotanical analysis and macrobotanical analysis. I'm going to do that here because the methods for collecting these are so vastly different. Microbotanical analysis, as the name suggests, is the study of botanical things that are too small to see with the human eye or the naked eye, or to see even with a regular microscope. Usually this requires some kind of specialized equipment to do these types of analysis. So the first is palynology, which is the study of pollen, ancient pollen. So all kinds of scientists study pollen because it's a very powerful tool for doing environmental reconstruction, something I'll talk about in a moment, in a few moments. There's also starch grain analysis, which is looking for these microscopic structures that exist in certain starchy vegetables, as the name suggests. And since starchy vegetables are often some of the plants that archaeologists are the most interested in because they're such important foodstuffs usually, starch grain analysis is really important. And then there's phytolithic analysis. These have all fun names. Phytoliths are cool. I think this is, by the way, I'm a macrobotanist. I don't get to do any of this stuff. I joke around all the time. My friends who are microbotanists, I talk about how they're like real scientists, because when you do microbotany, you have to like wear like a lab coat and goggles and stuff, because they work with oftentimes like really intense chemicals that can do terrible things to your body. For instance, with starch grain analysis, you have to use a chemical that burns away all of the inorganic material, which can include your bones, and phytoliths, you have to use an acid that burns away all the organic material, which is like everything else in your body, right? So you have to be really, they're kind of intensive processes. And in macrobotanicals, I don't have to deal with any of that, which is not something I feel particularly bad about. But phytolith analysis is cool because phytoliths are these they're literal fossils. They are little bits of silicate that get captured, collected up in plants as they live, and they fill in the interstices, the spaces between organic cells within the plant. And then as the plant dies and disintegrates and goes away, these little silicate masses can survive because they preserve much better than the rest of the plant. And then you can find these, and because they are this kind of negative of the cellular structures, they can oftentimes be identifiable to a specific species or genus, maybe of plant. And thus they can be used to identify the presence of plants that are no longer visibly present in a site. So again, these are quite cool, kind of newer analyses, relatively novel analyses in the last, maybe few decades. They've emerged and have become increasingly important. But traditional archaeobotany, the type I actually do, is called macrobotanical analysis. And as I said before, this doesn't mean we never use microscopes. We certainly do, because some seeds are very small, and charcoal has some identifiable structures that are microscopic, so we need to look at them under high-powered microscopes. But we're talking about remains from sites that are macroscopic. You can see them in a sample. And so this includes seed identification, right? So we find seeds like these wheat seeds you can see here on the right side of your screen. We can see them, and we can just simply have comparative collections and identification manuals to just figure out what they are. And then charcoal identification, which is trickier than seed identification, frankly. But charcoal is probably it's one of the most ubiquitous artifacts, period. It's certainly the most ubiquitous archaeobotanical material. It's just something we find on lots of archaeological sites, but it's just these chunks of charred wood. Who cares? Well, it turns out that you can actually identify them all the way down to species sometimes. And sometimes knowing, that comes from a red oak can be interesting. It can tell you certain things, but we'll get to that in our case studies. I'll talk a little bit about why that can be interesting. As I said before, these take, I'm going to talk about that in a second. And then, of course, we also have oral and written histories to help us. So as Lindsay said when she introduced me, I'm a historical archaeologist. I work in the 17th century mostly. And so most of the sites I work on have written histories. Those are not perfectly reliable. They're usually written by Euro-Americans, not always, but usually. Rarely written by Native Americans in this part of the world anyway. We also have oral traditions that can be passed down through the generations of both indigenous people and others. And these often have to do with plants. Plants are so important to culture, so important to the economy of most societies, especially agricultural ones, that people write about them, right? Tax records from ancient Egypt are all about how many bushels of such and such plants, and this is certainly true all the way up into in the United States during the colonial era. So sometimes we can rely on non-seed materials to learn about the botanical past. A little bit more about the methodology of how we do this. This is actually one of the most important aspects of any archaeology, but it's particularly important in archaeobotany and in our sort of sister subscience of zooarchaeology, which is the collection of animal bones that, as you'll see in our case studies, usually go hand in hand with archaeobotany. Work very closely with zooarchaeologists and archaeobotanists. Preservation is one of the big factors. It's like spatial context, right? Where in the world did an artifact come from? Taphonomy, which is a site formation process. How is a site made, and whether it was disturbed in the thousands of years since it was abandoned in the past or something like that, right? Along with those really important aspects that archaeologists spend tons of time understanding, another one is preservation. Every artifact that is found on an archaeological site is subject to its own preservational factors. Some artifacts have an frankly infinite level of preservation. Rocks, right? So if you've got a stone tool and I drop that stone tool on the ground today, in 200,000 years, it's going to look identical to how it looks today, pretty much, right? So does a stone last forever? No, of course it'll erode in millions of years, but in terms of archaeological scale, it is essentially as like infinite preservation. This is why stone tools are so important in archaeology, because they preserve really well. Ceramics too, right? Another really important artifact. They preserve really well, thousands of years, and they look just like they did the day when they were made. You could see people's fingerprints in them from creating them, right? It's really cool. Other artifacts do not preserve well. And probably botanicals are the worst preserving material. I mean, maybe along with like fabric, but fabric is frankly most of the time made out of botanicals too. Preserve very poorly. They only preserve, in fact, in what we call in the biz extraordinary preservational circumstances. And so that means they don't just preserve normally. They have to kind of have something something happened to them to make them preserve. The most common of these is charring. So most botanical materials are charred. So if you have seeds, like our wheat seeds from the previous page, if I go back to that, right, those are charred. You can tell because they're crystalline and black. So they were exposed to fire and they get charred. This makes them preserve because by being charred, they are no longer really edible. And so nothing eats them, right? And they don't get eaten by bacteria. They don't get eaten by small animals. And so they can survive sometimes. It also, of course, makes them very fragile, so they can easily be crushed, right? So we're just constantly worried about that. Another form of preservation is waterlogging. Some of the best preserved archaeobotanical materials in the world are from waterlogged sites. So these are sites that are literally underwater, and sometimes, especially if they're cold, botanical materials can preserve for very long periods of time in really, really nice condition if they're waterlogged. The last I would say is, there's more than this, but these three are the kind of the biggies, is desiccating. So this is the opposite of waterlogging. These are really dry environments. So like you're in the desert or a cave site, right? Some of the reasons these sites are so important is because they have really good preservation because they're so dry. Okay, so anything that sort of interrupts the breakdown of that object by natural processes. There's also sampling strategies, right? So because of these preservational factors, because we can never take it for granted that a botanical set of remains is like gotten there through normal circumstances, right? So stone tools, we could pretty much say, okay, these rocks are going to be similar to how they were 1,000 years ago. So we can kind of assume that there weren't tons more of these rocks that disappeared for some reason. All right, I don't want to say that doesn't ever happen, but normally we can kind of assume that. Botanical materials, we can't do this, right? Because they only preserve in these extraordinary circumstances, we have to be really careful about both how we collect them and how we integrate that data into our broader data set, which I'll get to in a second. So first of all, we have to sample to get archaeobotanical materials usually. So for macrobotanicals, this usually means subsampling. That means literally, if you find a hearth on a site, a very common practice is you'll bisect it, which means you cut it in half. So you get a nice profile drawing of it. Looks really pretty for your publication. And also then you'll take half of it and you'll bring that back to the lab and you'll float it, which is the thing you see on this picture here. This is actually a field flotation unit. Or this actually is water screening. They're doing water screening. That's not quite floated. It looks a little bit like this. But you float it, which means you submerge your whole sample, all the dirt, in water, and you agitate that water, and the botanical materials, because they're really light, will float to the top, and then you skim them off. That's called flotation. It's how we collect most macrobotanical material, right? But that's a very different strategy for collection than excavating a site and screening it in the field and just putting the stuff into plastic bags, right? So that means it's being collected in this very different way. Microbotanical materials, like pollen, often have to be collected through coring. So pollen, usually what you do is you go and do like a vibrocore down into a nearby lake bed. And so of course, those materials are not cultural. They're not directly from your site. They're not associated with the rest of your archaeological material. They just are environmental. You know, they came from nearby. You can date the layers of the core with radiocarbon dating, and so know what time period they came from, but they're not directly cultural. So the point I'm trying to get across is that we collect these materials in a really different way. So we have to be very careful about how we integrate that data into the rest of our data set. And something I'm not going to spend a ton of time on is we normally do this by using statistics. So we'll use either quite simple statistics or sometimes quite complex multivariate modeling statistics to integrate that data back into the broader data set that sort of accounts for the fact that there's not a one-to-one correlation between how a botanical material is collected and identified and how a different kind of artifact might be collected and identified. Right? So this adds a level of complexity, which is why this is a specialty instead of just something every archaeologist learns how to do. So that's something to sort of keep in mind. Okay, moving out of methods and into the interpretation, sort of latter aspect of what I wanted to talk about, is ultimately we're trying to answer questions, right? We're trying to ask questions and answer questions about past cultures. And so what kinds of questions can we ask and which can we answer? The first kind of umbrella category I would say are environmental. So was the environment different at a site than it is today? Is this, if the site's a few hundred years old or thousands of years old or tens of thousands of years old or a million years old, botanical material can sometimes tell us what that environment looked like. We call this environmental reconstruction. As I mentioned before, palynology is probably our best tool for this, but all botanical material is helpful for this. So for instance, the pollen core, you'll take this big thin core down through the earth, and then you take radiocarbon dates all up and down the core to tell exactly how old each layer is, and then you look at the pollen from near that radiocarbon date, and you reconstruct the environment, because the pollen is just a kind of passive collection of plant pollen from the whole environment, and you can use that to sort of say, okay, at this time period this was a thick deciduous forest, and then at this time period it was an open plain or whatever, and if we go earlier, it was like more like the Arctic or the subarctic, right? So environments change sometimes drastically over the course of archaeological timeframes. And so if we don't know that an environment is the subarctic and we're assuming it looks like it does today, we're going to be vastly off in our interpretations of what that site really looked like in the past. And then the second are more cultural evidences. This is more what I do with macrobotanicals, more directly cultural interpretations. And when I say we're doing cultural interpretations, I mean, this is when we're using botanical materials to do essentially what we do with any other kind of artifact. We figure out and assign value to the artifacts and the ecofacts, the botanical material, and we use that to make a determination about what it meant to live in that society, right? So examples like evidences of cultural interactions, cultural change and continuity, so how do cultures change or stay the same through time, and then ultimately this sort of big picture, big ticket questions like the influence of these big phenomena like colonialism, imperialism, globalism. All those things can be addressed with the botanical record, usually alongside other archaeological materials. They're most useful, like any archaeological material, in context with others. So like I said before, faunal material is often paired, but certain other kinds of artifacts as well. Okay, quick, crazy brief case studies. So I just want to take everything we just talked about, those methods, the types of archaeobotanical materials, and then the interpretive elements of this, to just give you some quick little like snippets of real studies. So these are all sites, as I said before, that either I've personally worked on or ones that I've had to research and read a lot about as a part of other projects. And so they're going to be pretty focused on Southern New England and Connecticut and from the historical period. Excuse my getting emails while I'm talking. So case study number one, I looked at, this was my master's thesis, so it was exclusively my project. I looked at these two sites specifically through a macrobotanical analysis as they relate to the boundaries of the Mashantucket Pequot Reservation in the 19th century. So these are both houses from the mid-1800s, 1830s to 50s or so. And they're very similar houses. The house constructions are similar. They were lived in for about the same amount of time. In many ways, these sites look very similar. But there's an important difference. 226 is located right in the heart of the Mashantucket Pequot Reservation. And I think during the 19th century, we don't have a great historical record for this, was inhabited by a Mashantucket Pequot family. 10244A nearby is outside of the reservation, and we know for sure that that site was occupied by a Euro-American family, a non-native family. And so that's the only, so controlling for all these factors, the difference between these two sites is one is a Native American site and one is a Euro-American site. And so what can the botanicals tell us about these sites? Like I said, the other artifacts all looked very similar to each other. The botanical materials look quite different, specifically the wood charcoal. So remember charcoal analysis, I said before, not the sexiest of archaeological remains, but actually were kind of cool in this case. So I looked at the charcoal remains from these two sites, and what I found was that the charcoal at 226, the Mashantucket Pequot site, was significantly better quality than the Euro-American site's charcoal. And this is in both categories, two categories: construction materials, so that's materials that clearly came from the house itself, and fuel materials. So this is materials that came from things like hearths and those kinds of features. And so what does this mean? Their interpretation of this is we can do it's an environmental reconstruction and also a cultural reconstruction. What we learn is, and this is backed up by historical documentation, is that the 1830s, 1850s were just about the nadir, the low point of forestation for Connecticut. Connecticut was probably 90% deforested at that time period. There was really a crisis. But the reservation may have been like a little refugia for higher quality, older stands of hardwoods. And this is backed up by some historical documents, because during this time period, the Pequots lodged several legal complaints that non-native people were sneaking onto the reservation and stealing firewood. So this seems to be backed up by the archaeological evidence. So again, kind of a neat little thing. Case study number 2. This is some work from my dissertation. Two, again, Pequot sites, but much earlier. These are sites from the 17th century. Kaluna Hill is a domestic village from the 1630s. Monhantic Fort is a larger domestic village from the 1670s. Both of them are Pequot villages. The pictures here are just kind of like little snippets that don't tell us too much. This is the house that I looked at from Monhantic Fort, the excavation of it, very complicated excavation. So what we've got here are two houses, two wigwams, one from the 1630s, one from the 1670s, both of them Pequot. Again, I did a kind of comparative analysis between these two in this part of my dissertation, and not particularly a botanical one, but botanicals were part of it. The broader patterns here are more interesting that include the botanicals. But this was a really interesting way to look at a period of a lot of tumultuous change for the Pequots. So in the 1630s, when Kaluna Hill is abated, the Pequots are a kind of independent tribe. They are in an active war against the English, which they are fighting to a stalemate at that point. By the 1670s, the Pequots had lost that war, called the Pequot War, and were put onto reservations by the English. And in the 1670s, they were actually fighting another conflict, King Philip's War, which probably many of you have heard of, very bloody conflict, where they were allied with the English. So the Pequots were allies with the English, and Monhantic Fort was used as a kind of starting point for allied maneuvers into Rhode Island to fight against other native communities, against the Narragansett and others. That's fascinating to me. These just short period of time, a generation or two, so much seems to have changed. What does the archaeology tell us? The archaeology tells us that there's a lot of complexity. Some things have stayed quite similar, and the food this seems to mostly be the case. The kind of food that we're finding at sites from the 1630s and the 1670s is largely the same. So diet hasn't changed very much, but many other things have. So the kinds of ceramics people are using, the metals people are using, the Pequots are using guns in the 1670s, and many other aspects have changed a lot. Architecture, however, like food, largely the same. The living in wigwams that look strikingly similar between the 1630s and the 1670s. So, this study has a ton more detail in it, but just to say that botanicals are one way we can look at this really complex set of change and continuity. Okay, case study number 3. These last two are going to go much faster. Looking at the site of Indian Town, also a Mashantucket Pequot site. This is a site from the 1700s, and this is not my research. This is from, you can see the publication on this from 1993. It was when I was 7 years old. So this is a little before my time, but done by my advisor at UConn, Kevin McBride, in that period. But I just think this is such a fascinating example of a use of botanical material along with faunal material to look at a really complicated historical question. And that is a question having to do with survivance strategies. So in the 18th century, this is now another half century after Monhantic Fort, the tribe is not really flourishing. Their demographic data is at just about the lowest it's ever going to get. The reservation is shrinking year over year because of encroachment and legal battles. It's a tough time on the reservation, and there's the tribe is facing enormous colonial pressures. And what you see on the reservation is two communities emerge that show two very distinct ways of surviving that colonial pressure. One community is kind of steadfastly refuses to convert to Christianity. They continue to live in wigwams, and what the food data tells us, the botanical and faunal data shows us, is that they are continuing to eat food that's got hundreds of years of tradition to it. Another community on the reservation, which is called Indian Town, we see people adopted Christianity, and we see a lot of other changes that go with this, the adoption of European-style houses, as well as big changes in diet that we can see in the botanical data for partially. And so this is, there's no judgment in this of which of these is a better survival strategy, but we just see that even within this one tribal community, there's really quite a lot of complexity and diversity in the ways that people want to try and I don't know, deal with these immense pressures that they were under to change or to alter their lifestyles or to simply go away or die out or whatever the broader culture wanted them to do at that time. So again, botanical is a really cool version of that. And the last thing I want to mention, just to say really briefly, just to show you that botanical material can also tell us about non-native sites in New England. Two other projects I have had a chance to work on as kind of an outside contractor doing botanical are these two Euro-American sites, also from the 1600s. So these sites are from about the 1640s or 50s, so a little later than Kaluna Hill, which I mentioned before. But these are explicitly Euro-American sites. The Waterman House site is in Marshfield, Massachusetts, was excavated by Past AHS, which is a CRM firm in Connecticut. They did a fantastic job. And then the Hollister site, which was excavated largely by the sadly late state archaeologist of Connecticut, Brian Jones, who some of you might have known, is a wonderful man who passed away much too young last year. I had a chance to work on the bots from both of these sites. And they are both amazing sites, incredible botanical preservation at both of them. And what they show us is that these Euro-American sites, despite there being tons of historical evidence from these early periods that Euro-Americans weren't supposed to interact with Native people, they were afraid of Native people, they didn't like their food, they didn't like their ways of life, and all these ways that they saw themselves as othered and separate from Native people, from indigenous people. What the botanical material and lots of the artifactual data too shows us is that does not seem to be true on the ground. These sites are just complex, complicated combinations of Euro-American made and indigenous made objects and traditional indigenous foods and European foods and plants and animals, all mixed together in really fascinating and entangled sort of ways. And so these are, again, botanicals that can be an avenue into understanding colonialism from both the Native American perspective and the Euro-American perspective during this time period. And that is all I've got. Thank you.
Well, thank you, Dr. Farley. So if people want to send me questions via the chat function, you can do that now. I am, as the moderator, going to start off with my own question.
Sure.
What site that you've either worked on or sort of been tangentially related to has had the most extreme differences between what we would see in the environment today and what you were seeing based on the botanical evidence?
At least in New England, that's usually a question of just pure age. So I've got, just as I said in my sort of intro, I've had a chance to work on some Paleo-Indian sites. So if those who don't know, the Paleo-Indian period is the earliest period of human occupation in New England. So some of the oldest sites, the oldest site in Connecticut is about 12,500 years old. So that's the kind of time period we're talking about, 12, 11-ish thousand years ago. Botanicals are very rarely preserved on these sites. And there's a handful of examples. And so when I say I've worked on these sites, I mean, I have friends who are paleo Indian archaeologists who find a single seed and they ask me if I can please identify it before they send it off for radiocarbon date, where it will be destroyed. And getting to identify a single seed from one of these sites is quite exciting because it's so rare that they show up. But those sites, it is really important to think about environmental reconstruction. And we do have great like paleontological data from those time periods. So we know we have a pretty good idea. Oh, there's still quite a lot of debate about it. But it would have certainly in Southern New England been much colder, much drier during that time period. But there's a lot of debate about whether humans at that time, Paleo-Indians were traveling and following caribou herds, which is the traditional interpretation of that period. That's largely based on this is getting into the weeds a little bit, but maybe for you, Lindsay, which is largely based on a kind of middle range theory, ethnographic analogy with subarctic regions in Canada. And so the environmental reconstruction says, oh, this would have looked a lot like a subarctic region. And subarctic people today, you know, or in the last couple hundred years, well, they hunt caribou and they follow caribou herds over these large spans of space. And so we think that's probably what Paleo-Indians were doing too. And the challenge in archaeology, this technique of using what's called ethnographic analogy, where you take a modern group of like hunter-gatherers and use them as a kind of model for something that might have been happening thousands of years ago. It's problematic and it's been critiqued.
Yeah, because it says like those people haven't changed, right, for today. Right?
It's really, exactly. There's a kind of undercurrent of that says, oh, these people who are living today, they're fully modern people living in 2020, are really just like people who lived 10,000 years ago. And that's problematic. But when you're talking about sites that are that old, it's hard to figure out how you theorize about what that life would have been like without using ethnographic analogy, which is why this continues to be a commonly used technique in archaeology. But the botanical data and the faunal data, which again are so rare on these sites, if you can find them, might actually be able to speak directly to that. I mean, do we really find caribou bones? Are we finding plant remains that people are eating that would have only existed in a subarctic kind of boreal forest or something? And the fact of the matter is that we so rarely found these materials, we still don't really know. And these couple of examples, chances where I've got to do this, you know, we find one seed and it's like, oh, it's a wetland, you know, grass or something like that. All right, well, those exist in tons of different environments. Doesn't tell you that much, right? So, but that would be, I would say, the type of sites where I've had to think the most about big environmental changes. Because if we're looking at sites from only a couple hundred years ago, well, the environment then probably wasn't that different than it is now. Again, not accounting necessarily for changes that are happening as we speak. That would be, yeah, I think that's my answer.
Hey, like I say, I love Paleo-Indian sites because I live in Ipswich with Bull Brook. So
yeah, Bull Brook's like one of the only sites where they've ever found any botanical materials.
Well, and did you read? For anyone who might not know about this, the Archaeological Conservancy just purchased the site of Bull Brook too to preserve it. So that was big news in my town. So
that's good to know.
Yeah, Ben Fort's working on that. So.
I mean, Bull Brook, for those who don't know, and I'm not an expert on the Paleo-Indian by any means, it's just one of the most important Paleo-Indian sites in the Northeast. It's one of the oldest. It's one of the best preserved. But keep an eye on a site that was just found in, what town was it in? It's up in northwestern Connecticut. The site is actually called the Brian Jones site. They named it after because he was working on that site when he passed away. And it's a 12,500-year-old Paleo-Indian site with really, it was like deeply, deeply buried in a floodplain of the Connecticut River. And so it's very well preserved. So there's going to be publications coming out about that in the next probably couple of years. So that I don't know who's going to do the botanical analysis on that, but
Well, let me get to the next question, but before I do want to plug, in November, our last speaker for the Diggin In series will be Jen Ort, who is affiliated with the Bull Brook 2 site in the Archaeological Conservancy. So if you're interested in that, sign up for that date's lecture. Okay, so I have a question from Kate. She says, hi, Bill, great presentation. And she wanted to ask about the forest cover in Connecticut during the mid-1800s. Where would you recommend looking for archaeobotanical evidence of the historical clearing between the 17th and 20th centuries. Or did you cover that in your thesis / article and she could look back?
Yeah, the best data I found for that was actually from it's called like the Harvard Forestry Project. And I think it's based out of Harvard. And they have collected every kind of evidence you could possibly collect to try and reconstruct forestry coverage in New England from pre-colonial period up through the 20th century. And they used, I know they used palynology, they used like dendrochronology and looking at standing trees, at current forest coverage, historical documentation. So they looked at like, there's like the Minor Diary is a famous diary from kind of the Groton, Connecticut area, where this guy is a farmer for like 60 years, every day wrote down what the weather was like and what, and he just took these incredible details about this stuff. And so from those, you can mine a tremendous amount of environmental reconstruction data, like day by day, was it raining in Groton? Like we know that for this 60-year period. And they compiled all of that to come up with these figures for forestry cover in New England throughout that period. And that's where I got that data from. They put it out in sort of a straight number where like, the forestry coverage would be between, 0 and 100%. And what you essentially see is this curve that looks like a very sharp curve from say 1600, where there's probably 80 to 90% forestation, to about the 1850s when it's at its low point where it's down to maybe 10% forestry coverage. And then it starts to, in the late 19th century, it starts to recover as agriculture becomes less and less prevalent in the Northeast. It's very easy to just track this with European style agriculture. And then actually nowadays we're back up to like, it's like in the 70s, I think, percent forested. So it's mostly recovered. Forests are not the same today as they were in say in the 16th century or something. But there has been a huge recovery since then. Basically because we don't do agriculture in the Northeast very much anymore. That's the major reason. So just clear cutting. People are clear cutting. And there's irony in that because I'm going on and on about this, but it's fascinating to me. There's irony about that because a lot of the English settlers, the explicit reason they come to New England is because of the wood. That's a very big selling point. The early colonists, when they're writing back to Europe to try and encourage investment and encourage people to come and settle in New England, they talk about these incredible forests that go on for miles and miles and miles, these beautiful old growth trees, because in England was basically deforested at that point. It was a crisis. There were, people just didn't have fuel wood anymore. So it was a huge selling point to get people to come to the New World, was actually this dense forest.
Excellent. Well, thank you. And I'm pretty sure Kate does not mind if you went on and on about that.
Yeah.
There is another question. Is there any indication of indigenous population numbers based on botanical findings?
Well, based on botanical, that is a very hotly debated question. What you might call sort of Native American demographics pre-colonially, and you hear figures that differ wildly from, there were, a couple of million Native Americans in all of North and South America, to there were 100 million, Native Americans in North and South America, which would have been a tremendously large number at that time period. Mostly that data is drawn, I think, from human remains studies. Like they look at demographic, like literal demographic data, like you just that's like from physical anthropologists who are looking at human remains from ancient burial grounds and things like that. But I'm trying to think. Let me think for a second how botanicals might help us. You could try and model, I guess, based on, well, when was agriculture adopted, and can we make some assumptions about population figures rising as agriculture is adopted? That gets tricky because in New England, agriculture is adopted in a very unusual way from the rest of North America. So in places like, Mesoamerica or Cahokia or something, right? Maize agriculture arrives and you get these huge civilizations, you get these huge cities, urban centers, and massive architecture, and these kind of Mesopotamian-like looking places. And not to make a direct comparison, they're obviously very different, but I just mean these large urbanization and population density. And this doesn't really happen in New England. Maize agriculture arrives here maybe 1,000 years ago, a little bit longer ago, and what the evidence mostly supports is that that life went on more or less as it did. People continued a fairly mobile lifestyle, and they integrated maize agriculture as a part of that.
Yeah.
I'm saying this in some ways in contradiction to a paper I published last year that looked at this question and sort of said the opposite, which we looked at architecture, and we said that architecture was actually changing a lot during that time. So it's complicated, was what I'll say. That yes, okay, over that 1,000 years, certain aspects of life seem to stay the same, others seem to change. What we found in this study was that architecture seems to change quite a lot. House size and shape changes quite a lot in that 1,000 years. And so I'm now far field of the point, but I'm mostly dodging the point that I don't know exactly how you would use botanicals to try and make that kind of an estimation on demographics. But I will say that it's a hotly debated topic of which there are no strong answers. Because of course, in the earliest days of colonialism, there were massive demographic losses. Who knows how many people died during those first decades where there's terrible virulent diseases that spread, and in some places populations were reduced by as much as 90% perhaps. And we don't know how widespread those were. We don't know exactly, we don't know a ton about those. So if that's true, if it was a 90% population density loss everywhere, then yeah, some of those really large demographic interpretations could be correct. It could have been many millions of people, but we don't know that for sure. Anyway.
No, that was great. So one last question before we let everyone go. Is it possible that wetland sites where there could be good archaeobotanical preservation, especially pollen, have been overlooked, primarily probably here in New England too, especially?
I would say yes. So it's true that wetland sites that just underwater archaeology in general, this is a whole field of archaeology, underwater archaeology, if you weren't aware.
Another Diggin In thing, we will have the Massachusetts State underwater archaeologist, Dave Robinson, joining us in October.
I have a minor in underwater archaeology in which I took several classes with Dave Robinson. He is fantastic. You guys should go to that. He's an amazing guy and so smart. And a great speaker. So definitely check that one out. I'll give a plug to both of those. Jen Orr too. She's brilliant as well. So definitely check those out. But yeah, so underwater sites in general, because doing underwater archaeology is challenging. There are very few underwater archaeologists, but some of the best preserved sites in the world are underwater sites and in wetland sites. So we can think of Monte Verde in Chile, or some of these sites like in Denmark where they have like whole canoes with their paddles perfectly preserved and all this kind of stuff. The botanical material that comes out of those sites is incredible, and you learn you learn a lot. So the question is, are there sites like that in New England? Probably. They're hard to find. Wetland sites are really hard to work on. Sometimes you can't work on them because they're kind of protected by environmental laws for good reasons. But they both are understudied, which I think is the question is implying, and I think that's correct. They're understudied compared to other landscapes, and they have potential for incredible preservation, which means we could learn a lot from them if we actually found sites like that. Like the Brian Jones site, that site was buried under, I don't know, like 3 meters of soil because it's in essentially a floodplain from the Connecticut River. It's essentially in a wetland. That's one of the best sites probably ever found in Connecticut, and we don't find those sites very often. So.
Okay, well, thank you, Dr. Farley, for speaking with us today, and thank you to everyone else for joining. Join us in 2 weeks for our own Dr. Suwana Selby Crowley, who will be speaking to us about the archaeogeology and her favorite subject, dirt. She'll be focusing on Seneca Village site located in New York Central Park, which was home to a predominantly land-owning African-American community in the 19th century. So sign up and join us in 2 weeks. And again, thank you, Dr. William Farley.
Thanks, everybody.